Okay, so let's get started. So hello everyone. Welcome to our today's Immuseum seminar. So it's my pleasure to welcome our today's speaker, Dr. Hye-Young Kim from Korea. So Hye-Young currently is a full professor in Seoul National University College of Medicine in Department of Medical Science. So Hye-Young actually completed his undergraduate study in Ewha Women University in study biology, and then continue his training in Seoul National University as a PhD student, study both genetics and also immunology. So after she complete his graduate training, she decided to move to US. She moved to Boston Children's Hospital, Harvard Medical School, and then did her postdoc training until 2013. From 2014, she completed her graduate postdoc training and then moved back to Korea as associate professor in Seoul National University College of Medicine. And then to 2021, she was promoted as a full professor. So Han-Young's lab is mainly interested in studying mucosal immunology, and then her lab basically interested in three different aspects in mucosal immunology, which including her lab studied the mechanism of allergic inflammation and asthma. His team's work have advanced our understanding of the cellular and molecular machinery underlying allergic inflammation and asthma. And then they actually identify different key pathways and molecules which drive the inflammatory responses in airways, leading to the development of more effective therapeutic strategies for managing asthma and the related disorders. The second part of her lab's work is mainly focused in innate lymphoid cells in immune regulation. Her lab extensively studied the role of ILCs in immune regulation, particularly focusing on their involvement in the lung disease and the systemic inflammation, and identifying different specific cytokines and transcription factors which regulate ILC function, and also discovery of a novel ILC subset. Last but not least, her lab also identified a novel neutrophil population on the mucosal surface, and then characterized these distinct subset of neutrophils which emerge in response to air pollution-induced lung damage, as well as renal fibrosis. So Ha-Young's lab is really, really productive. Based on her achievement, she actually got numerous awards and recognitions. I'm not gonna read through all the awards, but I want to just emphasize she was continually recognized as excellent research achievement professor from 2015, 2019, 2020 and 2022 in Seoul National University. And on top of that, she was also awarded as Blue Ribbon Awards on 2022, Minister Award from Ministry of Health and Welfare in Korea on 2023. And last year, she also was awarded as Seoul National University Hospital Academic Awards. So I think I'm gonna stop here. Thank you so much, Hye-Young, to deliver the lecture for us today, and we are very much looking forward to it.
Okay, thank you, John, for kind introduction and having me here. So I'm really honored to present my work in this immune gym seminar. So today, I'm going to discuss about innate lymphoid in aspect of airway inflammation. So as you know, where allergic disease are on the rise worldwide. So approximately 20% of adults affected this syndrome in Western country, and the prevalence of allergic disease is expected to increase up to 70% by 2050. So among these allergic disease, we are interested in asthma. So asthma is a chronic inflammatory condition of the airway. So if you don't have asthma, air flows smoothly through the airways. However, if you have asthma symptoms, several triggers like air pollution, pet dander, pollen, and other things can induce inflammation inside the airway. They should result in wheezing, coughing, shortness of breath, which is critical symptoms of asthma. Compared to other diseases, the mechanism of allergic inflammation is relatively well known. So when allergen encounter our body, this antigen-presenting cell capture this allergen and then present this allergen to T cell, especially this type 2 T cell. And this type 2 T cell produce IL-4, which induce isotype switching of B cell, and this B cell start to produce IgE, and IgE induce mast cell degranulation. And type 2 cell also produce IL-5, which induce eosinophil recruitment, and IL-4/13 from Th2 cell induce mucus production from epithelial cell, and they also induce airway hyperresponsiveness. So in terms of this allergic inflammation, T cells are very critical. So I'm not saying T cell is not important, but I want to say some different aspects of this immune response. So when you're using this leg deficient mice, which doesn't have any B cell and T cell population, these kind of mouse still develop inflammation without adaptive immunity. That means that there are other cell types can produce this cytokine. So in 2006, Andrew McKenzie group identified interesting population, immune cell population, and they call this cell type as a non-B, non-T cell. So as you can see here, their morphology looks like lymphocytes, but they don't express any Fc epsilon receptor, so they are not mast cell, basal kind of cell. But in response to this recombinant IL-20 type, this non-B, non-T cells start to produce large amount of IL-13 and 5. So they identified this unique population in 2006. And 4 years later, Koya's group in Riken, actually they found this natural helper cell in the PET-associated lymphoid cluster as indicated here. So if you see this section, there is cluster of immune cell, and actually Moro tried to identify this cell population, and she found that actually half of them are lineage negative, but express c-KIT and SCOUT, which is a stem cell marker. But this cell is distinguished from stem cell because this cell type expresses CD45 marker, and then this cell type also produces large amount of type 2 cytokine. So they reported this cell as a natural helper cell, and many other groups also reported similar populations. Andrew McKenzie group called this cell as a neocyte. David Artis group called this type of cell type as a multipotent progenitor cell, and many other groups called their own name like ID2 cell and so on. So researchers have decided to call unify the name of this cell as an innate lymphoid cell. So as you can see from their name, this cell type belongs to innate immunity, and their morphology looks like a lymphocyte. So the function of innate lymphoid cell is pretty similar to T cell. So T cell recognize antigen, specific antigen to activate this cell type, but innate lymphoid cell doesn't express any antigen-specific antigen recognizing receptor. So instead, they recognize some cytokine and environmental cue. As an innate counterpart of T helper cells, ILC can be divided into 3 different subsets like ILC 1, 2, 3. And this IL-12, 15, 18 can stimulate NK in ILC-1 to produce interferon gamma and TNF alpha. IL-25, 33, and TSLP stimulate ILC-2 cell produce these type 2 cytokine. And lastly, this iron beta and 23 can stimulate ILC-3 to produce IL-17 and GSS. So during my postdoc training, I discovered this innate lymphoid cell in the lung tissue. And then when we infect the influenza virus and challenged many different type of allergen, this type 2 innate lymphoid cells start to produce large amount of IL-5 and 13, and this type 2 cytokine induce glucose production and airway hyperresponsiveness. And we found that actually macrophage like alveolar macrophage and interstitial macrophage can produce IL-33, which stimulates this innate impulse in the lung tissue. In addition to our lab, many other groups also identified the role of innate impulse cell in terms of allergic asthma. So many other immune cells can interact with innate impulse cell in the lung tissue. But in the context of obesity-induced asthma phenotype, another subset of innate impulse cells are involved. So in this case, high-fat diet induced the NRF3 inflammation from M1 macrophage. And then this macrophage produces iron beta, which stimulates ILC3 to producing IL-17A. And this IL-17A directly induces the smooth muscle contraction around the airways. So although these innate impulse cells have been extensively studied in the mouse model, there are initial uncertainty about whether these cells exist in the human or whether they play a role in human asthma. So, in fact, the composition of innate impulse cells is quite different between mouse and human. So now we know the distribution of innate impulse in both spaces. For instance, if we look at this lung tissue, the major cell type in the lung tissue is this kappa-3 positive ILC2 cell. When I analyze innate impulse cell in the lung tissue, more than 90% of ILC is ILC2 cell. However, if we look at this human lung tissue, actually, when I check the human sputum sample from the lung, actually, I found that the portion of type 2 innate impulse is pretty small. So, given these differences, we need to consider whether findings from mouse study can be directly applied to humans. So we have to consider this point. But in 2014, Keslin actually identified this ILC2 cell from the peripheral blood from asthmatics. So they gated on this lineage negative population and then further separate this population as a CRTH2 positive, IL-7 receptor positive population. So this fraction 1 should be ILC2 cell and this fraction 2 should be other subset of ILC. And then they stimulate this fraction using IL-33 and they found that this IL-1 produces this type 2 cytokine, and when they're stimulated by IL-33, their size become very large. And then in combination with IL-33 and IL-25, this ILC start to produce type 2 cytokine like IL-5 and 13. And this is the interesting point. So when they get the blood from allergic rhinitis and allergic asthma, only the allergic asthma patients have a higher number of innate impulse cells in the blood. So they suggest that maybe the increased number of ILC2 cells in the blood can be used as a biomarker of this allergic asthma. And this is my data, although the number of samples is pretty low, because I collect the pulse fluid from the human asthmatics. And then combination with IL-27 iron beta, I can detect some lineage negative population start to produce IL-17A. So I guess this should be IL-3. So, but the sample number is pretty low here. So when I get back to Korea, I want to see whether this innate impulse is really important in the patient with asthma. So we recruit 18 healthy control and 50 asthmatics and try to figure out the role of innate impulse in this patient. So in Korea, actually, it's very hard to get a pulse fluid from asthmatic, but I think this pulse fluid can reflect lung environment better than blood. So we tried to get this induced sputum from the patient to a healthy control and try staining the immune cells here. But the total number of immune cells in the induced sputum is pretty low, so it's very challenging to staining this immune cell here. But when I see this population, I was happy we still detect some lymphocyte and myeloid population from this induced sputum. And then when we further separate, further stain this induced sputum using this lineage marker and IL-7 receptor, we can detect innate impulse cell population here. So compared to healthy control, asthmatics have a higher frequency of innate lymphoid cell induced sputum. And then we further divide this ILC population as a CRTH2 positive ILC2 cell, CK positive ILC3, and double negative ILC1, and check their frequency in the patients with asthma. And as you can see here, all the subset of ILCs are increased in the patients with asthma. So initially we expected maybe ILC2 or a certain subset of ILC should be increased in patients, but all the subsets are increased. We also check the macrophage in the induced sputum, and we compare healthy control asthmatic. And then again, we see that macrophages are increased, and it's 11C positive M1 macrophage and 206 positive M2 macrophage. Both of subsets are increased in asthmatics. So this result can be a bit disappointing because we expected that there might be specific subset of immune cells increasing in these patients, but we thought there might be some correlation between this increased cell type. So we conducted this correlation analysis. So when we conducted this analysis, we see the positive correlation between M1 macrophage in ILC1 and ILC3. In contrast, we only see the positive correlation between ILC2 cell and M2 macrophage. This result indicates that innate immune cells in the airway interact in directional manner, and which may influence the phenotype of asthma. So just the long story short, I found that actually eosinophilic asthmatics have a higher number of ILC2 cell and M2 macrophage, and by in vitro culture study, we identified that this ILC expressing this post-multi molecule can induce M2 polarization of alveolar macrophage, and this type of cytokine from ILC can induce this M2-like phenotype in alveolar macrophage. In the non-eosinophilic asthmatics, we have a higher frequency of ILC1 and 3, and the cytokine production from this ILC subset can induce M1-like polarization of the macrophage and neutrophilic inflammation. So as this study conducted in the patient with mild asthma, actually in the first study, it's hard to detect the correlation between ILC and decreased lung function. So in the following study, we collect samples from more severe patients. So here, when we check these indicators of lung function, our new code show significantly lower lung function. But there are some problems. So since we recruited more severe patients, many of them are actually smokers. So we have to divide our sample into non-smoker and smoker and analyze this full group. Again, we get the induced sputum from our healthy control and patient, and then compare the size subset again. Compared to healthy control asthmatics, we have a higher frequency of ILC, so we more focused on this difference between this non-smoker and smoker, and interestingly, only ILC3s are increased in smoking asthmatics. When we further divided this ILC population using this NCR marker, we see that the smokers have a higher frequency of NCR negative ILC3, which is known to produce IL-17. At that time, we also collect the peripheral blood and then try to check ILC population here, but as you can see, we cannot see big difference using this blood sample. So that's possible because the induced sputum may reflect more lung environment and blood is circulating there. But since we get this valuable sample, we want to see find more differences. And then at that time, we have a concept that ILC also have a memory-like characteristics like T cell. So I'll talk about this part a little bit later. So we have this idea. So we try to separate this ILC population using this memory marker of T cell. So naive T cell express a higher level of CD45RA, and memory T cell express a high level of CD45RO. So when using these two markers, actually our ILC population nicely separate to the RO positive ILC and RA positive ILC population. And then we analyzed our subset, and then we found that actually there is increase of this RO positive ILC3 population in the peripheral blood, and this ILC produce more IL-17A. So we expect maybe asthmatics have some memory like ILC population in peripheral blood. And we also compare this ILC frequency with the smoking amount. And as you can see here, ILC frequency in the sputum and peripheral blood show positive correlation with the smoking amount. So this result suggesting that maybe repeated exposure of smoke can induce the activation of innate lymphoid cell, and this cell may have this memory-like characteristics, although the mechanism is not known. And then in line with our idea, actually other groups also published a similar concept in the Science Immunology paper. So they found this CD45 RO positive ILC2 cells in the peripheral blood and nasal fluid, and they show that this memory marker expressing ILC2 cell have a higher mRNA expression of this type 2 cytokine. And then when we check the lung function with this frequency of ILC in the sputum and peripheral blood, we see negative correlation with the lung function and ILC frequency. So that means that if you have more ILC and you have more memory-like phenotype, your lung function will decrease. So they have some negative effect. So in this study, we found that actually cigarette smoke exposure induced the epithelial cell damage, and this damaged epithelial cell produced iron beta. And this iron beta and cigarette smoke together induce the CD45 RO expression on ILC3. And we call this as a memory-like ILC, and then this ILC produces higher level of IL-17A, and they recruit more neutropil, and they have negative correlation with lung function. So altogether, this mechanism involved in the asthma exacerbation. So I just showed two different patient cohorts, but actually asthma is quite heterogeneous disease. So people just focus on this type 2 high endotype of asthma, which involved in this T cell and eosinophils. However, there are many characteristics of asthma, like there is less T cells, less eosinophilic, and there are some steroid resistant patients, and there are some obesity-related asthma and pollutant-induced asthma. And then mostly this type of endotype is required this clinical, new clinical therapeutics. So we have interest in this steroid resistance because when these innate lymphoid cells are first discovered, researchers expected that maybe this cell type explain this steroid resistance. So people think we can control T cell using steroid, or this remaining cell may still respond. So indeed, this early study showing that there are increased number of innate lymphoid cells in steroid resistant patients. So this is CRS patient, and this is the paper showing that the nasal fluid steroid resistant CRS patient have a higher frequency of the CD45 IL-2 cell. But the opposite side of view also exists. So people also reported that ILC frequency and activity can be inhibited by use of glucocorticoid, and they also showing that T cells that stimulate ILC are sensitive to steroid. So this conflicting result lead us to develop the steroid resistant animal model of asthma and try to figure out what ILCs are really steroid resistant or steroid sensitive. So we developed this OVA and OVA 33-induced asthma model. So OVA is the standard protocol to induce asthma. So this OVA peptide can stimulate the type 2 T cell. And then when you induce asthma using this OVA peptide, these mice develop airway hyperresponsiveness pretty nicely. And this steroid treatment with dexamethasone significantly reduce the development of airway hyperresponsiveness. However, when you combine OVA with IL-33, which is supposed to stimulate IL-2 cell, this mice becomes steroid responsive. Dexamethasone cannot reduce the development of airway hyperresponsiveness. When we check the lung histology, we have a similar trend. So dexamethasone clearly abolishes the inflammation around the airway in OVA group. But if we look at this OVA and salicylate group, the inflammatory cells are remaining in the airways. So we check the cytokine production from T cell and ILC. So dexamethasone treatment can reduce type 2 cytokine production from T cell in both OVA and OVA salicylate group. But when you look at this cytokine production from this ILC, this dexamethasone treatment cannot reduce the cytokine production from ILC. Even in this OVA salicylate group, dexamethasone treated group show higher cytokine production in ILC population. So we think that the cytokine production from ILC may be responsible for the steroid response, and then we want to look at which pathways are involved in this mechanism, these responses. So we analyzed several pathways, and then we found that actually JAK-STAT pathways are upregulated in the steroid response ILC population. So we tried to in the in vitro study, we tried to induce this steroid response ILC, and so we combined several cytokines, and then we treated dexamethasone and figured out that the combination of the signaling and IL-33 signaling induced this steroid response of ILC. And more interestingly, when you combine this IL-27 and 33 all together, this IL-6 population starts to produce IL-17. So this is triple positive ILC become generated when we combine these three cytokines. So we called this population as a multipotent IL-6. And when you treated dexamethasone, which is steroid, they have no effect on this multipotent IL population. However, when you treated this JAK inhibitor, this JAK inhibitor clearly reduced this multiple cytokine production from IL-2. So we confirmed the role of this multipotent ILC by adaptive transfer experiment. So we generate this multipotent ILC and then transfer to red and common gamma deficient mice. We also treated the inhibitor here and look at their function. And adaptive transfer of multipotent ILC nicely induced the development of airway hyperresponsiveness and eosinophil recruitment. So this JAK3 inhibitor treatment can reduce the development of airway hyperresponsiveness as well as eosinophil infiltration into the parafluid. So go back to our initial model. We want to know the therapeutic effect of this JAK3 inhibitor. So we treated OVA and IL-33 together with mice, and then tried this test the effect of steroid and JAK inhibitor, and we combine this together. Actually, the single treatment of dexamethasone or JAK3 inhibitor have no effect on this combining model. So when you treated both dexamethasone and JAK3 inhibitor, it can nicely reduce the development of airway hyperresponsiveness as well as the inflammatory cell in the parafluid and the lung tissue. So we check the cytokine production from T cell and ILC again, and this dexamethasone treatment can reduce the type 2 cytokine production from the T cell, but they have no effect on ILC. It's opposite. JAK3 inhibitor have no effect on T cell cytokine production, but it can nicely reduce the cytokine production from ILC. So when you combine this together, we can regulate inflammation in the lung tissue. And more importantly, this JAK3 inhibitor clearly reduces multiple cytokine production from ILC. So in this story, we suggest that maybe steroid-sensitive asthmatics, they may have the T-cell dominant phenotype. So when you treat with steroid, it induces T-cell apoptosis and reduce cytokine production, so we can easily control asthma phenotype. However, in certain case, who has this mixed phenotype, like activated T-cell and IS population, steroid treatment may not be enough to regulate this condition. And actually, we found that in combination of IL-33 and this tech signaling can induce this multipotent IS population, which produces both type 2 and type 3 cytokine. And this multipotent IS becomes steroid resistant. So when you treat T-Jax3 inhibitor to block this signaling, we just have one signal. So steroid treatment can easily induce apoptosis of this IS population. And they altogether they can reduce the airway hyperresponsiveness and eosinophilic infiltration in the airway. So when we conducting this asthma study using mouse and human, actually, since we are studying innate lymphoid cell, we kind of have a hurdle because people think this innate lymphoid cell, these ILCs that belong to innate immune cell, and innate immune cell just involved in early responses. But as you know, this asthma is induced by repeated challenge of a certain allergen. So if these cells just belong to innate immunity, we cannot explain these cells are important in the development of asthma in humans. So that's always limitation of our study, and I have to think about this point. But this human study showing that IC3 have some memory-like phenotype, and they have a more active form. And other group also reported that in the CRS patient, they have CD45 autopositive IC2, and they have more cytokine production. So this characteristics is pretty similar to memory T cell. And then our last study showing that if these cells become memory phenotype, so they have a multiple cytokine production, they become a steroid response. So I think this concept is mixed, little bit mixed, but they go together. So we tried to look at whether IS have some memory phenotype. So when I share this idea with my colleague already doesn't like this concept because most of them are T-cell person. So, you know, you should not use this memory term in your IAC. So you just use train the immunity, that kind of thing. But today I just use this memory like, the term of memory like. So in context of T-cell memory, this characteristic refer to T-cell memory. So memory T-cell have long-lived and they have rapid responses. And this memory T-cell have immense proliferation and unique marker expression, and their location is really different, and they have functional heterogeneity and epigenetic modification involved in T-cell memory. So I try to figure out all this aspect in our memory like IC population. So this is unpublished data, so I just share my concept here. So we primed ILC just treating ILC the first time, and we rest ILC 1 month and re-stimulate ILC using this ILC and see whether this ILC have some memory-like phenotype. As you can see here, that the 33 treatment is potent stimulator of ILC2 cells. So this 33 alone can expand the ILC population in the lung, and the cytokine production is significantly increased. So when it rested 1 month, this mice and then re-stimulated IL-33 again, this IL-3 population become bigger. So it fit to around 50% in the lung tissue. It's quite a lot. And then the cytokine production from this IL-3 become even higher than its prime group. So it's like this character is similar to memory T cell. And then when you check the airway hyperresponsiveness, this second challenge group develop more airway hyperresponsiveness, and this inflammation around the airways become more severe than this priming group, and mucus production also increased in this second challenge. So we're wondering how long this activated ILC can be survived. So we just track the stimulate ILC and then track the survival by up to 3 months. And then unexpectedly, actually, this ILC survive longer than we expected. So it's long, ILC have a long-lived characteristic. And like our multipotent ILC study, when we stimulate ILC using this IL-33, this second challenge, the ILC start to produce IL-17. So this primed the first challenge ILC doesn't produce IL-17A, but when you re-stimulate ILC again, this 17 producing ILC start to emerge there. And then when you check the characteristics of the 17 positive population, you see that most of them are cathode positive ILC2 cells. So it's not from ILC3 and other cells. And we also check the location of innately improved cell using this lead-5 mice. So this 33 primed ILC proliferate, and then they located in parenchyma of lung tissue, as you can see here. It's stained in red color. And then when you check second challenge IL-33 again, you can see that this innately improved cell closer to this epithelial cell. So most of them are very near to epithelial cell line. So I think this stimulated ILC become closer to epithelial cell, and they can interact with epithelial cell more. And then lastly, we want to know whether this epigenetic control can regulate this memory like characteristics of ILC. So when you check the HTAG expression by bulk RNA-seq, this class 1 HTAG is increased in stimulated ILC. So we confirmed the HTAG expression using RT-PCR and Western blotting, and we found that actually HTAG 1, 2, and 3 and series are expressed in ILC population. So we tried to generate ILC specific HTAG 1 or 3 knockout mice, and then we checked that these mice show reduced HTAG 1 and 3 expression. And then again, we tried to generate the asthma model using this HTAG knockout mice. So we primed ILC using IL-33, and we rested 1 month, and second challenge with IL-33 again. And as you can see here, recombinant control increase ILC population and cytokine production. Or if we see this HTAG knockout mice, ILC frequencies are not increased, and cytokine production is reduced in this HTAG knockout ILC group. We also checked the airway hyperresponsiveness. It's reduced in HTAG knockout mice, and the inflammation around the airway is also reduced in HTAG knockout mice. The trend is pretty similar when you use this HTAG3 knockout mice. The frequency of ILC reduced, cytokines also reduced, and the development of airway hyperresponsiveness is reduced in HTAG3 knockout mice. So it's a still ongoing study, so we don't know exactly how this epigenetic modification regulates memory-like characteristics. So we suggest that this first stimulation enough to induce this effect ILCs or these ILCs produce large amount of IL-13 anti-radiation cells at the kind of thing that's enough to induce asthma. But the second stimulation even further induced the cytokine, and this second stimulated ILC start to produce a new type of cytokine like IL-17A, and this we call this previously multipotent ILC, but this become steroid response and they become more pathogenic, so they may involve the development of asthma phenotype. So in this study, we tried to figure out this characteristics of memory in T cell, and I would say that ILC also similar characteristics like the memory T cell. So in our study, we tested various forms of asthma using allergic inflammation and non-allergic inflammation. We also tested steroid-resistant asthma model. So in allergic asthma, IgG2 cell producing type 2 cytokine is critical, and they recruit the eosinophil and they activate M2 macrophages, so they result in lung inflammation and mucus production. But in non-allergic asthma, like obesity-induced asthma and airflow control-induced asthma, other subset of ILC may be important. And in our smoke study, we identified the CD45 RO-positive ILC is critical, and they interact with neutrophil. In the steroid-resistant forms of asthma, the combination of IL-33 signaling and JAK signaling pathway is required to this steroid resistant IS population. And then this population produced more cytokine, and we suggest that this steroid resistant IS also produce types of cytokine. Altogether, they may involve in more severe lung inflammation and airway hyperresponsiveness. Although I didn't bring the slide here, we also interested in other lung disease like emphysema and COPD, lung fibrosis. And then in this condition, in addition to ILC3 or other subsets are important. So we showing that ILC3 may be induced in this phenotype by interacting with the neutrophil. Other group also showing the ILC1 producing interferon gamma in this alveolar macrophage activation, and this alveolar macrophage produce proteins and NO that may involve in lung damage. So based on these findings, although most clinicians just focus on T cell, we suggest that ILC may be alternative possible target to treat allergic disease, especially ILC2 cells are extensively studied in mouse and humans. So targeting this ILC activity could be alternative target, but I would mention that in the case of a human, we have to consider another subset of ILC, like ILC3 and ILC1. So we're showing that activated ILC may involve different type of asthma, and we have to think about steroid resistance. Maybe ILCs are involved in this pathway. So I just start my slide here, and although we have our main interest is lung disease, like including asthma, COPD, and fibrosis. Recently, we have studied role of innate lymphosis in the developmental lung cancer, and we have studied innate lymphosis in many other diseases like CRS, steatosis, IBD, and lupus nephritis. So basically, we are interested in mucosal tissue immunity and innate lymphoid cell, and as Chan interested in the beginning, we identified the CBLF expressing neutrophil subset in context of B-cell particle-induced asthma. And then we also identified this role in lupus nephritis. So we tried to understand the innate immune network in various tissue and how this network regulates the development of various inflammatory diseases. Thank you for listening, and I would like thanks to Dr. Ji-Hyun Kim. She is now started her work in NIH from this month, so you may see her in the NIH. So she has conducted this asthma study pretty nicely, and I would like thanks to Jong-woo Han. He also involved in the cigarette smoking this asthma study. I also thanks to my all my passionate students who work hard with me. So thank you for listening. And if you have any questions or comments, I please to answer it. Thank you.